Over to you, Dr. Naweed.
Thank you, Zain, for that lovely introduction. And Bismillah ar-Rahman ar-Rahim. Assalamu alaikum to everyone, and especially Ramadan Mubarak to all of you. I know everybody's fasting, and after spending a long night during Taraweehs, people are still trying to join in. So I think I will try to really recapitulate, whatever I would have presented throughout the talk to make sure people can actually understand and join. So today, I think it's an interesting topic, and the topic really is that my lab, in addition to other aspects, it's really interested in understanding how anesthetics may affect brain development, brain plasticity, learning and memory, especially when younger children or the elderly are exposed to anesthetics. So really the overview of today's talk is that how brain circuits are put together during development, and then they communicate with each other through synapses, because all brain functions, from simple reflexes to complex motor patterns, learning and memory, they are all controlled by networks of brain cells, and these brain cells then communicate through specialized structures we call synapses. So then the question we will explore today is that how might various anesthetics affect neuronal communication, impact their viability, and alter their connectivity? And I will provide some very specific examples as well. And then the question is that because anesthetics are essential, we cannot perform any surgeries without them, as the pain itself could be more exacerbating than the effects of anesthetic. And similarly, pain, management is really important, because pain could also inflict a tremendous impact on our brain and the brain cells connectivity. So this is really the overview of today's talk. And the learning objectives today is to expose you to how brain cells communicate, meaning synaptic functions, how brain plasticity forms the basis of learning and memory, and how synapses are formed in the brain and modulated throughout life. The other important thing to remember is that we will explore maybe modes of actions of both local and inhalation anesthetics and their effects on both excitatory and inhibitory synaptic transmission. So the question really we will discuss in detail is, do anesthetics affect synaptic function? We know that they do, otherwise they wouldn't be able to block neuronal communication and wouldn't be able to put our brain to sleep. And whether they affect brain cells growth and synapse formation in a developing brain, it's really important to realize that the developing brain is highly plastic, where synaptic connections are forming, at an explosive rate, and their connectivity, is really relies very in an important way on how they communicate. And then we will look at anesthetic-induced cytotoxicity. Why is it important, and how one could mitigate those effects too? So as we know, the brain is fastest growing organ in a human body. There are hundreds of millions of cells added every 10 to 15 minutes. And moreover, there are trillions of synaptic connections that are forming in a baby's in utero. So when a baby is born, baby actually has twice the number of brain cells, but soon after birth, half of them are killed because of the programmed cell death. And once that happens, there will be no further addition of new synaptic connections or no new neurons till we die. So I think it's important to recognize is that the developmental patterns of brain are so robust and so fast that anything that could interfere that connectivity pattern will render the brain dysfunctional for the rest of life. Even though we know that there is a neuroplasticity, and these plastic changes are really important in terms of helping to regain that lost brain function through rewiring, but that rewiring is also limited to a greater extent. So when you think about brain, it's a structural and functional unit. We know that the neurons are the primary functional unit of brain cells. These neurons, there are tens of billions of brain cells in a human brain. Actually, we have more brain cells than there are stars in the Milky Way. And these brain cells are connected with each other through special structures. We call them synapses. So synapse is really a contact point between brain cells and different networks of brain cells. And we also know that the glia, which was originally thought to serve as a glue or a support system, also plays very important role in terms of really regulating the synaptic connectivity or synaptic actions. So when these synapses are functional, neurons are connected, they give rise to neuronal circuits, and these neuronal circuits really then are not hardwired, but rather they exhibit an enormous degree of synaptic plasticity. And this plasticity, meaning that the synapses can be altered, either augmented or depressed, forms the basis of all learning and memory in a human brain and also in the animals. So this synaptic plasticity then forms the basis of behavior, for example, from simple reflexes to complex motor patterns and learning and memory. And they really are in turns control all brain functions. We know that many of these aspects are regulated through genes. Genes define the initial patterns of connectivity, but our environment, which is the epigenetics, plays an enormous role in terms of reconfiguring those synaptic connections and modulating them and giving them the synaptic plasticity, which forms the basis for all learning and memory. So these are really the functional and the structural units of brain. This is what will be the focus for my today's talk today. So there are neuron to neuron synapses, whereby the neurons have extensive dendritic pattern, and they also have an axon. Dendrite receives the information from other brain cells, whereas the axon then communicates and passes that on to other neurons through special structures that we call synapses. So synapse is really the nexus at which all the information between brain cells are exchanged and transmitted. And then the glial cells also play an important role in terms of the synaptic connectivity and also modulating the synaptic connections, in addition to repairing the damaged brain cells or protecting brain cells from any infiltration from the immune system. So these are really the kernel fundamental units of brain. And we know that the genetic and environmental factors, they really play a very important role in shaping the brain connectivity. And these brain circuits, are changed all the time in our life. Whenever we learn something, our circuits are either altered, enhanced, and some circuits are really suppressed. So when you think about really development, we know that the developmental patterns of connectivity, whereby electrical activity of brain cells is really, really important. So we know that brain cells or neurons that fire together, they wire together. And if neurons fire apart, they fall apart. Similarly, we also know that the neurons that fire out of sync, they really fail to link. So during connectivity, whereas there are many molecular mechanisms that allow brain cells to recognize each other, but of all of these, the most important aspect is the electrical activity. And so cells that fire together, they wire together. And if this firing pattern is altered or perturbed one way or the other, we end up really neural circuits that are haywired or they are not properly connected. So this is really a take-home message, is that the neurons that fire together, they wire together. If they fire apart, they will fall apart. So neuronal activity plays a pivotal, important, essential role in defining the final patterns of connectivity in our brain. And this is really the important takeaway. And the examples for this would be, for example, if a kitty cat, you put a patch over one of its eye soon after its birth, just for a few days when these retinotactile pathways are developing, And when you remove that patch after a couple of days, this cat will be blind in that eye for life, because the sensory input that is coming in the form of electrical activity, which activate your photoreceptors and rods and cones, and then retinal ganglion cells. When these retinal ganglion cells receive that input, they discharge electrical impulses or action potentials, which then send a signal to your visual cortex. And that's how these visual cortex alignment will take place. And it's on basis of this electrical activity. Similarly, children who cannot hear, they actually cannot speak, because the firing of brain cells, going out of sync will make them fall apart and they will fail to connect. So this is really an important aspect of the entire talk. So the idea really is, in many cases, when you think about synapses and look at the overview of synapses, these synapses are specialized structures, and they are between pre and the postsynaptic neuron, whereby a neuron is connected to other cells through special structures we call synapses. So a synapse is really a functional, fundamental unit of nervous system. And if the synapses do not function properly or fail to establish, our brain is really completely out of its functionality. So the molecular composition defines the synaptic function, and we know the synaptic plasticity, which changes this molecular composition of synapses. It also influences synaptic function. So we know that all changes, which is adaptive, learning, memory, maladaptive changes, epilepsy, Alzheimer's diseases, they all owe their existence to the function or the dysfunction or the abnormal function of these synaptic connections. And we know that what happens is that during neuronal communication, we know that an action potential that is coming through the presynaptic terminal, it invades the axon. And when it comes in the area of axon, nerve terminal or synapses, it activates voltage-gated calcium channels. So when voltage-gated calcium channels open at the presynaptic terminal, it allows calcium to come into the cell. Calcium then binds to the synaptic vesicles that are carrying the packets of neurotransmitter and allows them to fuse at the synaptic terminal, whereby they are exocytosed or released, and they could bind either to ionotropic receptor, whereby the receptor directly gates the ion channel and it changes the membrane potential of the postsynaptic cell or the cell that received this information. There are other receptors which are metabotropic receptors. They are coupled with G protein coupled receptors, and most peptides actually function through those receptors, and their functions are a little bit slower. But not only that these changes occurring in the postsynaptic cell alter the activity of the postsynaptic cell, cause it either to be excited or inhibited, but a repeated stimulation will either enhance the efficacy of transmission or it will cause the depression of synapses. Moreover, it can also really bring about specific changes in the gene expression, and these gene expression changes then form the basis for long-term memory, which really is retained oftentimes for life. So it's really important the way the synapse function. And why am I telling you this thing? Because all anesthetics actually target this particular site in a neuronal connectivity pathway, whereby the local anesthetics often will block the action potential conduction by blocking sodium channels in the presynaptic site or in the presynaptic cell here. So when there is no action potential, there is no calcium influx, no transmitter release, no neuronal communication. And this is how the sedation is brought about. Our pain pathways are blocked. So this is something really important to note. The other thing to note is that anesthetics could also block the receptor function on the postsynaptic cell, thereby preventing the activation on the other side. Neurotransmitter release is also affected by a number of anesthetic agents, they block, and sometimes the reuptake of neurotransmitter back into the terminal is blocked. So these are the potential target sites for almost all of these anesthetics, and they will block either activity, they block conduction, or they will block release, or they will block the actual receptive end of a postsynaptic cell. So this is something really important to note. Now, when we think about synapses in the context of functionality, what happens is that during development, what we find is that GABA, which is most of the time is an inhibitory, a neurotransmitter, whereas glutamate is the excitatory neurotransmitter. Anesthetics will either block the excitatory neurotransmission or they augment or enhance the inhibitory synaptic transmission. This is how they function. They will either block excitation or they will augment inhibition to bring about depression in neuronal synaptic connections and connectivity. But what happens during development is that GABA, which in the adult is inhibitory, is actually, and it gates chloride conductance. In a developing brain, GABA itself turns out to be an excitatory neurotransmitter. And so, it's really important that when we are using anesthetics, even if they are supposed to, for example, block GABA receptors or enhance GABAergic synaptic transmission, which is inhibitory, but during development, GABA itself will be excitatory. So if we used anesthetics to block any conduction or brain activity or brain functionality, we realize that it is also blocking excitatory synaptic transmission that is mediated by GABA, which would otherwise in the adult brain have been excitatory. So both excitatory and inhibitory synaptic connections will be suppressed by anesthetics if they are used in a developing brain. So this is really important takeaway. And we also know that it's very difficult to really study these processes in the intact brain. How do we know that brain cells that actually fire together, they wire together? And what happens if the activity is blocked? Because you are using drugs in a whole intact brain that will block activity all over and not any other specific connections or connected sites. So what we do in our lab is that we use a variety of brain chip interfacing technologies where we can grow large networks of brain cells on a semiconductor chip, and we could apply various anesthetics at various stages of their development and ask the question, what might be the effect of these anesthetics on these growing neurons? We can also explore whether anesthetic exposure of early developing neurons affects their cell viability, their growth, or their connectivity patterns. And of course, this is done in cell culture, but what we can also do is that we can use the whole brain slices, whereby we can actually take the intact brain slice in its entirety, we can put it on a chip and record from intact neuronal circuit. So it's really important that after development that proceeded normally, if you block that electrical activity or you prevent the enrichment or the sensory feedback input to come into brain, we realize that the brain is seriously compromised. So epigenetic factors, you're looking at 2 brain scans, and you look at the normal brain of a 3 or 4 years old child, you can see that how densely packed it is. Whereas a child who went through abuse, extreme neglect, was not allowed to interact and was kept and chained, their brain development is seriously compromised because they were not provided with synaptic inputs or the visual input or the epigenetic environmental factors that would have allowed their brain to shape and function like a normal brain. So you could see the size of the brain is significantly compromised. And also you can see that the both left and right side of the brain are really not in sync or connected with each other. So we know that anything that perturbs activity, even after birth, will render the brain dysfunctional for life. And these are really something very, very important to remember. So the question is that, when you induce a state of anesthesia, which is really used to block, of course, or suppress, transiently suppress synaptic activity or brain cell functionality. We block neuronal activity and synaptic connectivity. This is how we bring about sedation in the human brain prior to surgery, and also for pain management later in when they are suffering some kind of a pain. So the modes of really action and the potential target sites of are often very difficult to decipher in the intact brain. And that's why a number of cell culture of the isolated brain slice preparations are used to test how anesthetics block synaptic transmission, where they act, and what are both the short and long-term effects of blocking this connectivity, particularly in a developing or in an adult brain. So here is some anecdotal evidence. We know from a large number of studies and animal models that blocking synaptic connectivity during early development renders the brain circuits compromised. The animal exhibit learning and memory deficits when they are older. This is a given. Also, I think we know in humans, the evidence is anecdotal. It's still equivocal. There is no really absolute evidence to suggest that it compromises the connectivity, but there is ample plausible evidence to suggest that anesthetics do indeed affect brain connectivity. And when they tested on children or were exposed when they were 8 years of age, and when they test their cognitive functions later in life, they find that those functions, some aspects of brain's cognitive functions vis-a-vis learning and memory are compromised. So this is really take home. So when we were the first team to actually demonstrate way back in early 90s that anesthetics really have detrimental effect on brain connectivity, and a series of studies that were conducted by us and others has now made our Canada health agencies to put labels on all anesthetics when they are given either to pregnant women or when they are given to young children. So this is really the take home, and this is what we will test the effects, whether it does or it does not affect brain cell connectivity. Of course, we know that these synaptic connections that are formed, they are really, very important during connectivity, and brain cells will function as I showed you earlier, and an action potential comes, invades the terminal, invokes the calcium influx, calcium enters the cell where it binds to the vesicles. Vesicles then are really released, and repeated stimulation of a particular terminal will cause greater amount of release, but it also primes the synapse. So these vesicles then get docked at the terminal. So when after the tetanus or after a repeated stimulation, when you fire the next action potential, these vesicles are ready to go and they get hammered, and you get an augmented response, which forms the basis of synaptic plasticity or short-term memory. So this is something really important takeaway, that these neurotransmitter receptors and the potential synaptic sites are the targets of all anesthetics that are used. And we know that they could be, a number of proteins that have to get to the synaptic terminal to allow them to function. On the presynaptic side, you have synaptophysin, synaptotegamin, synaptobrevin. These synaptic proteins are kept in vesicles, and they contain the contents of neurotransmitters. And on the other side, you really have postsynaptic density protein, which really brings about all the postsynaptic machinery, and it tethers it, and it puts it right in front of the synaptic terminal. So when an action potential comes in, transmitter release occurs, and then the postsynaptic response is elicited. And these are the two important proteins that I'll be talking about. But it's really important to know that because synaptic transmission forms the basis of neuronal communication, be that within the brain circuits or from brain to periphery and periphery to brain, where the pain sensations brought about, most anesthetics, be that local or inhalation anesthetics or general anesthetics, target all of these potential sites whereby the synaptic transmission is altered. So it could be the neurotransmitters like glutamate, GABA, dopamine, serotonin, acetylcholine, and many of these transmitters and their release machinery is altered in response to anesthetic treatment. So when you think about a single action potential in a simplistic way for most young students, a single action potential can produce one-for-one excitatory postsynaptic potential in the postsynaptic cell, and they're fairly consistent. We keep the postsynaptic cell below threshold so it doesn't fire. So you only get an excitatory postsynaptic potential, which involves release of transmitter. But if you fire a burst or a tetanus, you get a much bigger response. And if you come back and fire the same action potential, you can see that the first action potential is significantly potentiated, and this forms the basis for short-term memory. We have published a number of studies showing that anesthetics actually do not affect short-term plasticity or short-term memory. And these are all published studies, so I won't dwell on these today for very long. So what we can actually do is using a semiconductor chip, rather than recording from 1 or 2 cells, we can grow large networks of these brain cells at different sites and different electrodes. And we can test a variety of neurotransmitter, a variety of anesthetics, both local, which could be, propofol, and we have used lidocaine. We used many of these different transmitters, different anesthetics, local and inhalation anesthetic, to find out how they affect synaptic transmission and at what level. And these are also published studies, so I wouldn't really dwell. We can monitor the activities of up to thousands of neurons concurrently under very highly specialized and controlled conditions. And of course, those are cultured neurons. You could say that they are not really the intact, tissue. We can also prepare brain slices whereby we can isolate a brain slice, specifically record from the area where learning and memory occurs in hippocampus, and expose them to various anesthetics. And you can see that the activity patterns of these neurons could be monitored with exquisite accuracy, and we could determine how and for how long these anesthetics affect neuronal synaptic transmission. So I won't go into the detail. We have published a number of studies showing how anesthetics specifically affect synaptic transmission in the brain circuits. So this is something to really remember. So the rationale for this particular talk and study really is, we know, and the logic of inquiry is that we know that since blocking neuronal activity in a developing brain renders connectivity haywire, meaning that the activity is all messed up, so it therefore stands to reason that anesthetics and pain management drugs that block activity, they will also impact brain connectivity. So this really is the rationale. So if a pregnant mother, later stages or even early stages of development, had to undergo surgery and she was exposed to anesthetics for a longer period of time for some reason, a developing fetus will have brain activity in addition to her own activity blocked. And if the electrical activity is blocked in these developing brains, what would be the impact of brain connectivity? I'll give you an example. For example, when you have these premature neonates, babies, most of the time nurses will evaluate, guess what the level of their discomfort or pain might be. And they are often given morphine. They are often given other pain-killing drugs. And even though those receptors may not be developed yet, so the actual threshold for morphine application or giving them is really very, very high. So if you block that activity, and you will find that these children will often have cognitive deficit and learning and memory difficulties throughout their life. Similarly, younger children that are exposed to anesthetics because of one reason or the other, and I must really reiterate here that we cannot perform surgeries. So it's a necessity to use anesthetics. We have no way out because pain itself could be exacerbating and could have even worse impact on brain connectivity, a persistent pain or chronic pain. And so we have no choice really to other than to use anesthetics during surgery. But the question is, it's really important for us to find out and make an effort to find out what happens if we have repeated exposure to anesthetic of a developing brain in a young child or in a young, developing brain. What might be the effect of blocking this activity on brain connectivity or brain cells, cell death or their growth patterns in a developing brain? So in the end, once we know what are the potential targets, where lies the really target of these anesthetics, one could go back and design strategies to help mitigate. And also, if we know, the sites at which these anesthetics are specifically targeting their cytotoxic effects in the pathway of learning and memory and cognitive function, we can design strategies that will enrichment strategies targeted to be able to specifically regain that lost brain function and enhance, really augment brain cells activity and functionality if we know exactly. So that's really is the rationale. And the question that we asked, that do anesthetics really affect neuronal viability, meaning the cell death or their activity? We know the activity is affected, synaptic function and connectivity. These are all prerequisite for healthy brain development. As I told you earlier, cells that fire together, they wire together. So the general hypothesis is there's the anesthetic-induced perturbation of neuronal activity or function renders the brain connectivity dysfunctional. So we will test this hypothesis and see what happens. So the idea really is that if a young baby or a young child is exposed to anesthetic, even repairing congenital heart problems, other defects that they may have, other abnormalities. So they are exposed to anesthetics during early stages, and these anesthetics block the activity which was prerequisite for connectivity. What happens to the adult brain when these kids actually are grown up? So today I will really talk about two of these transmitters that are extensively now being used at our children's hospital and other places too, specifically for children. And one of them is sevoflurane. Sevoflurane is an inhalation anesthetic. And recently, as a supplement, people have started using dexmedetomidine, and dexmedetomidine turns out to be less toxic. And it also has been shown by us and others that it mitigates the toxic effects caused or induced by sevoflurane, desflurane, and other inhalation anesthetics, including propofol, ketamine, and other neurotransmitter as well. So I think the idea would be not to put these anesthetics to shame because we cannot function without them, but to find really strategies that will mitigate these long-term cytotoxic effects of anesthetics on a developing brain. It's also important, I think most people who are working in a clinical setting, if they are dealing with elderly patients who showed a cognitive decline in their later stages and needed a knee replacement or a hip joint replacement or any other major surgery, and it is really very well documented that their cognitive function declines and they have delirium at the end when they recover. And oftentimes they never recover their cognitive function back to where they were. So anesthetic in the elderly, where the synaptic connections are already formed, but their continuous functionality requires, really it's important, requires persistent activity. And if anesthetics block that persistent activity on those vulnerable synapses that were degenerating or undergoing degeneration during neurodegenerative diseases, for example, Alzheimer's, these anesthetics will hit them hard as well. So the elderly will also have delirium and cognitive dysfunction because they were exposed to these anesthetics. So, it's really important for us to be able to really find out what are the potential target sites and how could we really mitigate. So in our experiments, our experimental protocols is twofold. Today I will talk about three major sets of experiments that we do. We can take actually, day 0 newborn pups, and we can slice the brains out, either hippocampus area or the cortical neurons, dissociate them in airtight chambers. We grow these neurons. Once they are really allowed to settle down for a few hours, we use various concentrations of anesthetic and are using a hospital grade anesthetic machine, which delivers anesthetic, which is really the concentration, a clinically relevant concentration, a concentration that the actual cerebrospinal fluid would have of these anesthetics during a complete state of anesthesia. And we also can then pre-treat them or inject them or expose them using dexmedetomidine at various different concentration as an adjuvant to these anesthetics, because most of the time these anesthetics are now being both local as well as inhalation are being used in conjunction and get together. And then what we do is that we will do live and dead cell assay. We will look at the Ethereum homodimer 1, which is an indicator of cell death. So when a cell dies, it will pick up the dye in the nucleus, and this is how we can recognize the dead cells. And then we can also label live cells, urine calcinin AM, and this will indicate healthy and living cells. So this is the first aspect. We ask the question, when we expose these neurons to anesthetics, what will happen to their live dead assay or their viability? The second is that those cells that do survive, we then ask the question whether the extent of their growth or neurite growth is affected by these transmitters following the initial exposure. And then what we do is that as a connectivity marker, we will use synaptophysin on the presynaptic site as to really identify presynaptic puncta. And then on the postsynaptic site, we stain for protein, postsynaptic protein or postsynaptic density protein 95. And then we could see these juxtaposition of pre and postsynaptic neurons coming together as an indicator of synaptic puncta or synaptic connectivity. And these puncta are then counted to determine whether anesthetic affects one particular type of synaptic connection or the other. And then what we also do is that we will then do mitochondrial morphology, because mitochondria, we have shown, is the one that are affected by anesthetics most. Mitochondria is a powerhouse. It provides the energy. And in normal conditions, you will see them fused. These mitochondria are fused together and they work together to be able to really provide energy that is needed for all cellular functions. And then you will also see some intermediate mitochondria where some of them are, go through fragmentation process to be transported to other areas, but then they will fuse again to form a very healthy mitochondria. And the fragmented mitochondria is that when they are all dispersed all over the place and they do not function as one functional unit. So think about them as you have multiple engines around your, Karachi Express or Tezgam or whatever train it is that is pulling together. But if you remove one or the two of the engines, it becomes really hard for one engine to drive or pull the entire, all the bogies. And then we look at the, of course, mitochondria fragmentation will result into production of free radicals. The free radical cause oxidative DNA damage. We also look at the reactive oxygen species, because these reactive oxygen species can affect both the cellular viability and the kill cells, or their growth, or their connectivity pattern. So this is really the simple protocols that we use for this first assay. And the other approach we use is that we will take these young pups and we will inject in them, expose them, either the newborn pups, to sevoflurane once or twice, or we use different concentrations, either 1.6, which is used clinically, or 3.2, which is also used clinically. And then we allow these pups to become adults, so within 2 to 4 months, and then we test their behavior, whereas the controls receive the control solutions, which is, a simple either air perfused through or a saline injection. And then what we do is that once these animals become adults, they have only been exposed to these anesthetics either once or twice, and maybe for shorter or longer time period. We will then do Morris water maze test, whereby we allow the animals to really look for, in an open environment, to look for the platforms that they are. So animals will swim around and they will then locate their platform. We also do novel object recognition test, where the animal is allowed to habituate for 15 minutes, for 2 consecutive days. They become familiar. Then we put, 2 objects, novel objects, and the animal can recognize those objects. And then what we do is that we could either replace the older object that animal had familiarized with a novel object, or we move those, both of those, or one of these familiarized object away. And then finally, we do the open field testing, whereby the animals are really allowed to explore their environment, and we would like to see which particular quadrant the animal will spend most of the time, and whether they have the exploratory behavior or not. And the final really is the contextual field conditioning. The animals are put habituate, allowed to acclimatize, and for 2 minutes. They are shocked given 3 shocks, 1 every 2 minutes. And when they are brought back in the day 2, they freeze and they will not move around. They will fear that this is an environment where they get shocked. So these are really the tests that we perform on those animals to test the behavioral, their outcome in response to early life exposure to anesthetics. And what we really find is that when we use, for example, desflurane, which was also extensively used in the past, and still may be used for in certain medical colleges or hospitals. I don't know why it keeps flipping. We then look at these neurons and look for live dead assay. The red dots actually determine the dead cells that picked up the dye, whereas all the green dye is picked up by healthy cells. And as you can see that in control, we really see no cell death when these cells are dissociated, then exposed to anesthetic. Sevoflurane shows some dead cells, but desflurane was really the one where we saw the maximum cell death that occurs in response to clinically relevant concentration, where the sevoflurane was 1.3% and desflurane was 4.3%, and they were both exposed for 1 hour. So the really question, again, the viability is that the desflurane causes a more extensive death, whereas sevoflurane, under those in a comparative range, did not cause cell death. Then we looked for the neurite extension. And what we also find is that whereas in control neurons extend extensive dendritic and axonal branches, whereas in sevoflurane, we see some curtailment of these branches, but in desflurane, the neurons actually are very limited. They don't branch out, they extend longer processes, but the branching is also concerned. So what it really suggests is that, desflurane as compared to both sevoflurane and control affects, both of these inhalation affect neurite quality of the growth and the extent of the total growth. And these data are summarized here. The total length of brain cells that are growing as compared to control, we find is that both sevoflurane and desflurane curtail the growth of these brain cells in a developing paradigm or a developing system. And then we also looked at really the total length of growth, and we find that also in the actual 2 days and 8 days, the growth is significantly retarded, both in the presence of sevoflurane and desflurane. It's interesting that desflurane initially, the actual total length is much greater, but because they don't branch, 1 or 2 branches that emanate, they will extend their processes, but then eventually the controls catch up, and then we see a slowing or less extensive branching patterns of these cells. So then we looked at the synaptic protein. So I've shown you so far that indeed a number of anesthetics affect the actual cellular viability. So in a developing brain, early stages when brain cells are differentiating, proliferating, migrating, you can imagine that all of these parameters will be affected if the brain is exposed to anesthetic at that stage. And then we looked at synaptic connectivity when we look at postsynaptic density protein and then synaptophysin, and the merger is the one that really shows both of these juxtaposed against each other, and it shows the number of synaptic connections. We count these botons in both normal and in the presence of sevoflurane and in the presence of desflurane. And you can see that as compared to really a control, both sevoflurane and desflurane have fewer puncta of both the synaptophysin as well as the postsynaptic density protein. So the take-home message is that it does indeed reduce the expression of synaptic proteins in these cases. And these data are summarized here. The localization of synaptic terminals is really significantly impacted by both desflurane and sevoflurane. They really prevent formation or juxtaposition of synaptic terminals between these neurons. And then we also asked the question about what will happen, what are the potential underlying mechanisms or target sites that these anesthetics are really affecting. And so one of the things that literature suggested in our own work, we asked the question whether anesthetics are somehow affecting the energy store of these neurons, which is required for both cell survival and growth and connectivity. And the common denominator may be the mitochondria. So we looked at actually mitochondrial fragmentation, and what we find is in the control, we have extensive mitochondria that are fused and they're very healthy and rich, whereas sevoflurane and desflurane both, the mitochondria starts to go in number, they become fragmented, and they are not a cohesive unit like a train or a bogie in a bogies of train connected together. And perhaps this may be the underlying mechanism that the anesthetics are really working. So in summary, sevoflurane and desflurane, they inhibit neurite outgrowth and they cause neuronal damage in premature neuronal cultures. Sevoflurane and desflurane, they compromise the expression of synaptic protein, which then renders the synaptic network dysfunctional. And then finally, sevoflurane and desflurane mediated cytotoxicity most likely involves the mitochondrial structure and function. So that really is the question, and that study was published. We really wanted to now go back and ask the question, how could we mitigate this, sevoflurane-induced cytotoxicity in these cultured neurons? So we started another series of independent experiments. We thought that if dexmedetomidine is used, or neurons are pre-exposed to dexmedetomidine, will it mitigate sevoflurane-induced cytotoxicity? So that really was the question. But we first needed to test whether dexmedetomidine itself, affects, for example, any of the parameters that we are going to examine. For example, cell death, growth of neurons, and also connectivity. So when we use 1 micromolar dexmedetomidine, we do see some cell death, where the red dots are indicators on from day 3 to day 7. And then what we use the 10 micromolar, we see extensive cell death. So in other words, higher concentration of dexmedetomidine are also cytotoxic, but these are really not clinically used concentrations. But nevertheless, if the neurons are exposed to higher concentrations, one would find that their survival is compromised. And these data are summarized. I think in the interest of time, I will just give you a take-home message. So then we asked, does dexmedetomidine itself on its own affect or compromise neuronal growth? And when we look at the actual control cells, what we do find is that it actually, interestingly, it affects growth in a different manner. It really causes, 1 micromolar will cause more branching, but the extent of total neurite outgrowth is really, compromised in a way. But they have more branching patterns, and these cells will grow more rather than control, even grow more than or really control where they elaborate branching patterns. So it does not affect, in other words, the growth of the cell. And then we asked the question, what about synaptic puncta and the synaptic density for presynaptic and postsynaptic? And we also find that dexmedetomidine on its own, when it's used on both cortical and hippocampal neurons, in culture, it does not really affect puncta, whereby pre and postsynaptic sites come juxtaposition, and the computer software counts number of these co-localized punctas of pre and postsynaptic cells and tells us the data is that the dexmedetomidine does not significantly affect the actual puncta or the synaptic boutons between these cells. So this really gave us the confidence that it in its own right, dexmedetomidine is not neurotoxic. Now the question was, can it now prevent the sevoflurane-induced cytotoxic effects? And also, I think we examined the mitochondria with dexmedetomidine. It does not affect the mitochondrial fragmentation. The mitochondria were really healthy, and they were all in a big, mesh of train-like structures, very healthy in functionality. So all the parameters that we use to determine whether anesthetics affected cellular viability, growth, connectivity, and mitochondrial fragmentation, dexmedetomidine treatment did not affect any of those parameters. So this gave us confidence that, okay, we can really go ahead and see if dexmedetomidine could mitigate sevoflurane-induced cytotoxicity if the cultures are pre-treated with dexmedetomidine. So again, this sevoflurane exposure resulted in increased cell death, as I showed you earlier. And then what we did was that we asked the question whether it also causes mitochondrial fragmentation, free radical production. And as I showed you in this set of experiments, we also found that it increased the fragmented mitochondria. And then we asked the question, now, can we look at really fragmentation, whether this fragmentation of mitochondria induced by sevoflurane causes the production of free radicals, a reactive oxygen species, which in turns could affect cell viability and cell death, and if these effects could be mitigated by dexmedetomidine. And the answer is really yes, indeed, dexmedetomidine could impact or affect if we pre-treat. Dex pre-treatment can rescue sevoflurane-induced cell death and also reverse mitochondrial fragmentation. So this was really interesting and exciting, that now you have a mechanism or a tool that could mitigate sevoflurane-induced cytotoxicity. So the next question was, what happens to actually, does anesthetic exposure of the neonates affect learning and memory when tested in their adulthood? So yeah, as I told you earlier, pups are born, we expose them to anesthetics, inhalation anesthetic in a container, either once or twice, and then we put them back in their normal environment with their mother, where they're healthy, they feed well, and they are locomoting, and we monitor their growth. And after 2 months, we then test their learning and memory. And what we find is that, interesting aspects, which has never been reported before because the changes were so subtle. We found that the sevoflurane exposure, it really increased the anxiety, and it reduced, animals' willingness to explore in an open field environment. So the data is actually published, and you can go and search, but I will give you just a take-home message in the interest of time. If you look at the control animals, they will go all 4 quadrants. They explore their environment, and they spend considerable time in all of these environments. If you expose them to 1.6% sevoflurane for 1 hour, one-time exposure, we find that they're still okay and they still continue to be able to explore all quadrants. But if we, expose them twice with same concentration of sevoflurane, 1.6%, what we find is the animals become restricted to one or the other quadrant, and they no longer have the courage or the exploratory behavior that will allow them to explore all 4 quadrants. So this reluctance or unwillingness to explore in an open field environment tells us that something that we need to keep an eye on these children, whether their inquisitiveness and their willingness to explore and their courage to be able to really take on tasks is really somehow compromised by sevoflurane exposure. And one-time exposure, it still is okay, there is enough plasticity, but if they have to undergo repeated exposure to anesthetic for one reason or the other, we know that it will reduce their willingness to explore, or really render their exploratory behavior compromised in an open field testing. We also really noted something which is really very interesting, and it has never been reported before, is that the sevoflurane exposure results in anxious and erratic and indecisive behavior. So if these animals were exposed earlier, soon after their birth, maybe once or twice to anesthetics, these animals were anxious. So they show the activity patterns which is reminiscent or similar to what you see in hyperactivity syndrome or ADHD. They were erratic and they were indecisive in their behavior in terms of really looking.
Sir, the mic is closed. Not here.
Can you hear now?
Yeah.
Okay, so let me go back. I think these bars are often confusing. Anyways. So what happens is that when we expose, these animals, pups to, they resulted in anxious and erratic and indecisive behavior, they would still be able to really explore, but in a sense that the latency for their responsiveness was really significantly decreased. And they had erratic behavior. The animal exposed to 1.6, they will run around all over the place and often really aimlessly exhibit what we call an anxious and erratic behavior, in most of these cases. So sevoflurane also exposure compromised their recognition memory. So the animals were really not able to recognize the object or pick up the object in a first go, even though they did find the object, or their targets on site, but it took them much longer. But because they were erratic behavior, they were just swimming around, they will still hit the target, but the sevoflurane also compromised their recognition memory, because most of their ability to find the target was erratic and stochastic, rather than very organized and targeted. So we also then asked the question, could all of these parameters be reversed if we pre-treated these animals with dexmedetomidine? And when dexmedetomidine pre-treatment, we found that it decreased the anxiety and increased animals' willingness to explore during an open field testing. So this is really remarkable. This is very interesting and very reassuring that one could use this particular anesthetic agent to mitigate anesthetic-induced toxicity. And what we actually find is that when they were pre-exposed to dexmedetomidine, animals were in all 4 quadrants, and they were really having an exploratory behavior, and they were willing to explore and go out, rather than the animals that were hit by 1.6% sevoflurane twice. Those animals were not very exploratory, and they were not willing to explore in an open field testing environment. So this is really something interesting. And then, last few couple of slides, we know that the dex pre-treatment also produces less anxious, less erratic, and more decisive swimming patterns. So dexmedetomidine also mitigated these, the anxiousness, the erratic attitude, the behavior of these animals, and then indecisive swimming patterns that once they were exposed to dex or pre-treated with dexmedetomidine. So finally, what we are also asked the question, because these animals were exposed earlier, we tested them 2 months later, there must be some epigenetic or genetic changes in their protein composition or in the makeup of their brain structures that may have been hit or compromised by these anesthetic treatments. So we looked at really gene expression patterns of these cells. So these animals were exposed, and after their behavioral testing, the animals that were coded and labeled, then we sacrificed those animals. We isolated their brain's hippocampus area, and then we did a nanostring study whereby we looked at close to 770 genes that may have been differentially altered or expressed, either upregulated or downregulated in response to either sevoflurane or dexmedetomidine. So this is something really an interesting experiment. When we looked at it, we found that there were actually several genes that were altered. Sevoflurane and dexflurane, they differentially modified gene expression patterns in a large number of these different genes. And we picked those genes, and it turns out that many of these genes, they are really linked to a number of cellular functions, and these are either downregulated or they're upregulated. And often when we use both dex and sevoflurane, we see that the gene and expression patterns actually are very similar. So genes that are altered by sevoflurane can be really rectified, or their expression patterns could be prevented when we pre-treat them with dexmedetomidine. And most of these, we looked at most of these genes, and they were either highly upregulated, many fold, or downregulated significantly. We went and we looked at many of these genes as to what they were. And then we found out that majority of these genes are either involved in learning and memory, which is neural plasticity, and some of them in cell survival, which is in circadian rhythm, which is the NAPS8, NAPS2, and is BMAL1, axonal migration, which is another gene autophagy and apoptosis, so cell death. Some of them are involved in cell death. Some are involved in synaptic plasticity, neurogenesis, and also myelination in many cases. So most of these genes are really related, very specific to cell function, survival, cellular migration, and axonal growth, and also in synaptic plasticity. And finally, I would like to really show you that in addition to these, many of these genes which are involved in cell death, they are also upregulated in response to anesthetic or sevoflurane exposure. And they cause cell death and demyelination, oligodendrocyte precursor, and the axon growth, cell death, myelination. Also, myelination is really important because, again, if the axons are not myelinated, their conduction will be slower or perturbed. And similarly, neurogenesis. So a number of these genes regulate lots of functions, and we are now testing all of these genes one by one to determine, either in a knockout animal specifically, to see if we could generate a phenotype that is really reminiscent of what happens after sevoflurane treatment. So in other words, the take-home message is that the number of genes that are involved in mitochondrial function, they also induce cell death, they can induce fragmentation of mitochondria, and then also they could really affect the memory retrieval, decision-making, recognition, memory deficit, and also anxiety. And all of these parameters, when we saw that the animals were anxious, they had really recognition, memory was compromised, they were reluctant to explore their environment, the decision-making, which is the prefrontal cortex part of the brain, that many of these functions are perturbed or altered. And these are long-term changes which really need to be mitigated in the longer run. So a number of these papers have recently been published, and you can actually look at the data more carefully, whereby we have demonstrated that indeed these anesthetics cause, and now there is a flurry of activity even in the human cases where people are coming, the anesthetists are coming forward and they are really looking at examining it in more carefully. So these papers are also published very recently. Now, a couple of really brilliant and bright students who really worked on this, and particularly Nuria here, she did her PhD in my lab in less than 2 years, and now she is in Stanford doing her postdoc. Shadab Batul was also a student from University of Karachi. She finished her PhD. So they all graduated at the same time. There were 4 of these students who did their PhD. I'm really proud of them. So Nuria is the one who actually did most of this work. And I'll be happy to take any questions that you might have.